So sorry, I was interrupted. So I was saying there's a higher diversity of ammonoid, and each of the four families of ammonoid has its own time span. So we will look at that in the next slide. For now, what you need to know about the distribution of ammonoid is where they are found in South Africa. So what is interesting is that they are found in two different basins in South Africa, two basins that are close to the sea, the seashores, the Algoa Basin and the Zululand Basin. And the two basins are dated to the Cretaceous and have been correlated thanks to their ammonoid content. So the correlation of these two basins was done well before the dating method were available, and it was done using the content in ammonoid fossils. So this is, for example, an ammonoid from Port Elizabeth, and you find the same species in the Algoa Basin and in the Zululand Basin. So they are very, very useful for correlation. And this is why, because all four families have different time spans. So the most primitive family of ammonoid has the most simple suture. So it's more complex than the nautiloid type of suture, but it's still a very simple type of suture, which is S-shaped. So you can see it's a zigzagging. It's a very simple zigzagging suture, a little more complex than the nautiloid type of suture. And this type of suture characterize the goniatids that are found in the Devonian and Permian. So here it's an example of a goniatid from Morocco. So you do find goniatids or goniatites. I'm not sure how you should pronounce that. I would say goniatids. Let's go for goniatids. So these goniatids are found in the Devonian and in the Permian. So they are typical Paleozoic ammonoids. So they lived alongside the orthoceres. Then you have a little more derived, the ceratites. So the ceratites, instead of having S-shaped suture, they have a spoon-shaped suture. You can see it's very conspicuous on that one. Here, the spoon-shaped suture, U-shaped suture, if you prefer also. So, and this is very typical of ceratite. They have a little more complex suture. They are a little more derived, and they are found in the Triassic. So right after the goniatids become extinct, you have the ceratites that take over in the Triassic. And then you have the proper ammonites. So the ammonites are your typical ammonoids, and they were extremely abundant, extremely diverse, and we cannot tackle all that diversity right now. The thing that you should know is that they have a very highly complex suture line. Highly complex suture line. So you can see I mean, it's difficult to describe that suture. It looks like a plant, if you want. It's very, very ramified. It's very, very branching. It's very, very complex. So this type of suture is directly derived from the spoon-shaped suture of ceratites, and they started living in the Triassic right after the goniatites become extinct, and they become extinct at the end of the Cretaceous during the Cretaceous Tertiary boundary, which is also the time when dinosaurs went extinct. So this is why on your geological time chart, the one I gave you in the during the first lesson, the Mesozoic is marked as the dinosaur and ammonite time. It's because the two typical fossils that you would find there are ammonites in the sea environment, and dinosaurs on the land, on the ground environment. And one type of ammonite that is very interesting for correlating Cretaceous basins like the KwaZulu-Natal and the Algoa basins are the heteromorphs ammonites. So they are like the ammonites. They have a very complex suture lines, as you can see here. But unlike the ammonites, they are not completely coiled. So some of them look like, well, I'm not sure what this is supposed to look like, a saxophone. So they look like a trumpet, like this. So Miloceras from KwaZulu-Natal, and some of them, like that Bochianites from the Algoa Basin, are completely straight. This is an ammonite. This is the suture. So the suture line testifies the complexity of the suture lines, shows you, demonstrates that it's an ammonite, but it's completely uncoiled. So just like an orthocere, basically. So that's why when you find something, a cephalopod that has a straight shell, you have to look at the suture line. Otherwise, you may identify an ammonite for an orthocere or vice versa. So watch out for the suture line. If the suture line is simple, it's a nautiloid. And if it's straight, if the shell is straight, it's an orthocere or orthocerid. And if the shell is coiled, it's a nautiloid. It's a nautilid. And if you have a complex suture, it's an ammonite. And depending on the complexity, it can be a primitive ammonite like a goniatite, or a more derived ammonite, a proper ammonite with a complex suture line. And then if it's very, very derived, in the Cretaceous, the very last surviving ammonite evolved an uncoiled shell, so very similar to the orthocerids. So that's why you have to watch for those suture lines. First thing you look when you want to identify the shell of a cephalopod is the suture line. Look for the suture line. Okay, last group of cephalopods, the belemnites. So the belemnites, we are not going to spend too much time on them because they have a very simple morphology. What you will find of a belemnite is this. So the bullet-like thing, very dense, very robust, very strong, very resistant to erosion. That bullet-like shape is actually what we call the rostrum. So the rostrum was the top of the shell of the squid-like organism. So that squid-like organism had a shell, and that shell has a small thing on the top of its shell, which is called the rostrum. That's actually something that you find in cuttlefish. So if you go on to the seashore, if you go on the beach, you will find some cuttlefish things that are called the cuttlefish bone. It's actually the shell, so that red part here. And you will see that this shell has some lines, and these lines are the suture lines between the part of the shell. But that's not very important because that's never preserved in the belemnites. What is preserved is the rostrum, that tiny spiky thing at the end. So in belemnites, it was very, very big and very, very dense. And that's why you find it preserved. So, and again, the ecology is the same as the other cephalopods. It's an open sea predators. They are open sea predators. The time span is interesting because it overlaps with the ammonites. So Triassic to Cretaceous, they also went extinct with the dinosaurs. So it's a purely Mesozoic fossil. If you find the belemnite, if you find that bullet-like shape, you know you're looking at the Mesozoic. So now be careful, watch out. We are still in the mollusks, but we changed. We are no longer looking at cephalopods. We are looking at gastropods. So your gastropods are your snails, basically. So the gastropods, they are like the cephalopods. They have only one valve, and it's spiraled, so it's coiled, if you prefer, but it's coiled in a conical way. So it forms a cone, a conical shape like this, or like this, if you prefer. So that's the big difference with the cephalopod. Your gastropods, your snails, have a conical shaped shell, and also there's no septa, there's no sutures in gastropods because there's only one chamber. So all of this is one cavity inside the gastropod shell. And the gastropods are quite different from your cephalopods because some of them are herbivorous. They are not all carnivorous. They can live on sea. They can live on land. Think about your regular snail that you see every day. So they can live on sea, they can live on land, and they can live in fresh water. For example, the ram's horn snail lives in fresh water. And you have marine gastropods, and you have, like this, land snails. So these ones are fossils from the Pliocene of the Western Cape, and they testify that this sediment that you see around, they demonstrate that this sediment around was formed under continental conditions. It was a land sediment, so probably during a flood or whatever. But yeah, this is not a sea environment. And gastropods are not very useful for dating because their time span go from the Cambrian to present. So they originate during the Cambrian explosion, but they don't show much difference since then. So the very, very interesting part about gastropod is really the paleo environment. Like depending on the seashell you're looking at, it will be a marine gastropod like this one or a land dwelling gastropod like this one. We change again and we go to the bivalves. So mollusks, yes. Bivalves, yes. So we are no longer looking at the gastropods or at the cephalopods. It's again a different whole family of mollusks. The bivalves or bivalvia. So that's your regular clam, pectens, mussel, oysters, your regular seashells with two valves. That's the big difference. They have two valves, so you can see that's a modern one, and here you can see that's a fossil one from the Devonian of South Africa, from the Cederberg area, and you can see the two valves are symmetrical. That's the big character of bivalves. The two valves are the same. You can see they are the mirror of each other. The two valves are symmetrical, and if you look at your bivalve from the front, from the mouth, if I may say, if you consider that the two valves together form a mouth, if you look at the mouth, you can see there is a horizontal suture, there's a horizontal symmetrical axis between the two valves. So the axis of symmetry runs between the two valves. If I'm telling you this now, it's because we will see another kind of seashell that has a different type of symmetry, of course. So keep this in the corner of your head, in the corner of your brain, horizontal symmetry, because it's going to come back. So the bivalves are typically marine species, although you will find some of them living in a continental environment. Some of them live in rivers or in lakes. You have mussels like freshwater mussel, for example, and I think this one is a freshwater actually species. So you have different types of environments reflected by your bivalves, and they can live in any temperature. They usually live fixed. They don't move, although some of them are capable of moving, like the pectens, for example, but usually they live fixed. They don't move during their lives. And as I was saying, with the oyster, you have different types of environment, of energy, that are reflected by the thickness of the shell. So the thicker, like mussels live have a thinner shell, and they live in a less disturbed environment than the oyster. And the time span of bivalves. So what's interesting about the bivalves is that they are quite rare during the Paleozoic. Here you have a counterexample, by the way, Devonians from South Africa. but usually they are quite rare during the Paleozoic, but they become very abundant in the Mesozoic, and they are extremely abundant since then until now. So even now you can find a lot of bivalves if you go on the beach. So if you find the bivalves, you know you have more chance to be in the Mesozoic than in the Paleozoic, but as I was saying, there are exceptions. And actually the reason why this time span is like this, it's because there is another group of invertebrates that is dominant in the Paleozoic and that becomes rare in the Mesozoic. So they have the opposite distribution in time than the bivalves, and they look very much like bivalves, and it's the brachiopods. So the brachiopoda, they are not even mollusks. They are a group on their own. They are not mollusks, but like the bivalves, they have two valves. And the only way to differentiate them is to look at the symmetry. So they are not symmetrical. Oops, a little typo here. They are not symmetrical. So in the brachiopods, you have two valves, like in the bivalves, but in brachiopods, the two valves are not the same. You can see here and there. Unlike bivalves, the two valves are different, and the axis of symmetry doesn't run horizontally between the two valves, it runs vertically through the two valves. So the axis of symmetry runs in the middle of the animal instead of in between the two valves. And this is due to the fact that one of the valves in brachiopods has this tiny hole, that opening for the pedicle. And they use that pedicle. That pedicle is basically a foot, so you can call it a foot instead of a pedicle if you want. So you have a pedicle valve, a valve that bears a foot, and that foot grips is attached to the ground. So it can be attached to a rock. So they usually live in a high energy environment. And in this high energy environment, they can remain attached to their substrate using that foot. So that's why the two valves are not the same. It's because one of them has the foot on it and not the other. And that's why you have an axis of symmetry running through the two valves instead of in between the valves like in bivalves. So this is a very, very, very important distinction that you need to know. It's always in the exam. That's a classical example of things that always end up in the exam. So you really have to know your difference between the brachiopods and the bivalves. The ecology is very similar to the bivalves, so that's not where you will find the much interest of the brachiopods. As I was saying, the interest of brachiopods is that they are highly dominant in the Paleozoic, and then in the Mesozoic they become rare up to the point that they are almost completely extinct today. So today you still have a bunch of surviving brachiopods like the lingula, and what's interesting about the lingula is that it lives only in brackish water. So I showed you earlier, like I think it was yesterday, I showed you the fossil of a lingula from the Devonian of South Africa, and it is a marker of brackish water. So usually estuaries or lagoons or stuff like that. So lingula is very interesting in terms of ecology, and it's one of the few surviving brachiopods today. But in the Paleozoic, and particularly in the Devonian, they were very, very abundant. Like you can see that rock that is covered with those winged brachiopods that are called spirifer. So you can see the axis of symmetry is there, and you have one wing and two wing. Here, axis of symmetry, one wing, two wings. So it's very, very distinctive. The symmetry of the shell is extremely distinctive in brachiopods. So now we change completely of invertebrate group. So we were looking at the mollusk, we were looking at the brachiopods, and now we look at the arthropods. So the arthropods are your typical creepy crawlies, your typical rojas, like the insects, the shongololos, the scorpions, the dung beetles and stuff like that. So the arthropods are your typical land invertebrates, land-dwelling invertebrates, and particularly the insects, because you can see on this graph that the insects actually constitute more than half the diversity of animal species, well, more than half the diversity of species on Earth. So here you have the plants, here you have the bacteria, here you have the fungi, and here you have the vertebrates. Where are they? The vertebrates are there. So things with a backbone are all contained here. It's 40,000 species. There are millions and millions of insects. Millions and millions of insect species. So they completely dominate biodiversity. So insects only. No, we are not talking about all the arthropods. So it's important to talk about them. So in arthropods, you have an exoskeleton, but this exoskeleton is not one valve or two valves. It's many segments. It's made of many segments that are articulated to each other. So think about your shongololo. Well, that's not a shongololo, that's a centipede actually, but it looks similar enough to a shongololo, and you can imagine that the shongololo is like many parts that are articulated together, and the tiny legs that are articulated to these parts, and this thing move because each part is articulated with the next one. So that's typical of arthropods. So the body is articulated, the mouth parts are articulated, the legs are articulated. Actually, the mouth parts are modified legs in arthropods. There's probably between 3 and 10 million species living today, and there's just not enough human beings on Earth to count them all. So there is a fair bit of uncertainty on the actual number of species, but it's probably between 3 and 10 million. Some people even say 30 million species. So that's how much we don't know about how many species are living today. And the ecology of arthropods is very diverse. You find some arthropods in the seas, like the crustaceans. You find some arthropods on lands, like the insects. So it's very diverse, but usually you won't find them in cold environments. So you find arthropods in mild or hot environments, and mostly tropical environments. And the time span is as the other invertebrates. They originate in the Cambrian explosion, and they are still alive today. So the first group of arthropod, and probably the most important one in terms of paleontology, is the trilobites. So the trilobites are dead now, they don't exist anymore, but they were the dominant arthropods throughout the whole Paleozoic. So that's why I call the Paleozoic the age of trilobites. It's because they dominate completely the ecosystems during the Paleozoic. You cannot walk on Paleozoic sediments without finding trilobites. So they are very typical of Paleozoic. And they are very easy to identify, which makes them very good index fossil. They are easy to identify because their name, trilobites, comes from the fact that their body is divided in 3 lobes. 3 lobes, trilobites. 3 lobes, one on the left, one on the right, and one in the middle. And you can see very clearly on that example from the Devonian of Prince Albert in South Africa. 1 lobe, 2 lobe, 3 lobes. Very easy. And if you cannot see that, you can see the division in 3 segments. So a head, a body, and a tail. So the head is called the cephalon, the body is called the thorax, and the tail is called the pygidium. And the head has a kind of a nose that is called the glabella. So the cephalon is the whole part here, and that part is the middle, is the glabella. So the trilobites, they are divided in three. 1, 2, 3 lobes, 1, 2, 3 segments. Very easy. And they are usually bottom dwellers in the seas, so they are typical of a usually shallow water environment. And as I was saying, they are typical of the Paleozoic, and they are very, very abundant in the Paleozoic. Another group of interesting arthropod is the chelicerate or chelicerata, where you find your spiders, scorpions, and the very fun and weird horseshoe crab. That's a horseshoe crab. You've probably never seen that before. It's a very funny type of arthropod, but it's a typical chelicerate. Why? Because it has only two segments. It has an abdomen here, and it has a cephalothorax here. So the cephalothorax is the fusion of the head, cephalo, and the body, thorax. So cephalothorax. So if you find, and it's very typical of chelicerate, so your spiders also doesn't have a head, it has a cephalothorax. The head is fused to the body. So if you look carefully at a spider, you will see that the legs are directly branching out of the head. And if you look at the scorpion, it's the same. The legs of the scorpions are directly coming out of its head. So it's very fun to, once you've noticed, you can only see this. It's the only thing you see. So and it's very, very typical of chelicerata. And the chelicerata have chelicers, which are the most part, and they are usually turned into pincers in chelicerata. So you can see here, in the horseshoe crab, you can see the pincers. You know the scorpions have pincers, so the spiders are the only exception, actually. They are the only one without pincers. But if you find pincers, you're almost sure that you're looking at a chelicerata, except if it's a crab, of course. You know your crabs. Crabs are not chelicerata. They are crustaceans, of course. So the chelicerata are interesting because they are found in the sea, like the horseshoe crab, or on land, like your spiders and your scorpion. And in South Africa, we have a very, very good fossil record of chelicerates, and that's why I'm bringing them up. So we have the oldest terrestrial scorpion in the world, found in the Carboniferous near Grahamstown, and you can see it's the pincer, so the mouth part, the chelicerates. So we have the pincer of the scorpion that indicates a terrestrial environment. And we have sea scorpions, so or pseudoscorpions. Sometimes they are also called pseudoscorpions. They are sea scorpions called the Eurypterida or Eurypterids that lived in the Ordovician to the Permian. So these are very interesting index fossil because they are typical of quite a narrow part of the Paleozoic. And in sea scorpions, you have the typical chelicerate organization of the body with the head bearing the legs, so the head body part, the cephalothorax, and then the abdomen coming after that. And you even have the tip of the tail, which is the telson, which will become the dart in scorpions. And the other groups of modern arthropods, we can go through them very quickly, but you already know them certainly. So the crustaceans, like the crabs and the lobsters, they are very easily identifiable because they usually have large antennae and they have a shell that is reinforced with calcium carbonate, much more than other arthropods. For example, insects, myriapods, and scorpions and spiders don't have calcium carbonate in their shell. So the crustaceans are quite unique for that. They usually live in the seas, though there are some species living in fresh water, like your ostracods. This is an example, so you can, if you're interested in knowing more about ostracods, you can Google that. And the same as any other invertebrate, they originate in the Cambrian, and they live to the present. So in general, crustaceans are not very interesting for biostratigraphy, but the ostracods particularly are very used for biostratigraphy because their shell, you can see their shell here, which is bean-shaped, varies quite a lot in between species and in time. The insects are very easy to identify because they have wings. So of course, they are the only invertebrates with wings, so they are very easy to identify. You have two examples here, an insect wing from the Jurassic of South Africa, and here a complete fossil insect from the Cretaceous of Botswana. And you probably already know that they have three pair of legs instead of 8 in the spiders. So that's why spiders are not insects. It's because of the number of legs. Also, the insects have 3 parts in their body. They have a head separated from the thorax, separated from the abdomen, unlike the chelicerates. So the head and the thorax are separated in insects. It is not in chelicerate. Insects are mostly found on land, though many of them have a freshwater larvae, but usually insects mark a continental environment, so it's interesting for paleoecology, and their time span starts from the Devonian to modern days. So of course there are still a lot of insects living today, but the first insects are from the Devonian. And the final one, the myriapods or chongololos, or your centipedes, they are quite easily recognizable because their body is very long and it's the same segment that repeats many times with, oh sorry, not eggs, with legs. So it's the same repeated part. And same as insect, they are typical of a continental environment. So if you find a fossil myriapod, you know you're looking at continental environment, and their fossil records start in the Silurian. I think it's time for a break.